Hello everyone, I'm Qichang Zhao, a PhD student from the School of Computer Science and Engineering at Central South University. I'm honored to have been invited to speak at this conference. Today, I would like to present my paper, DDAffinity, predicting the changes in binding affinity of multiple point mutations using protein 3D structure. My presentation will cover the following aspects. Mutations play a crucial role in natural evolution by disrupting protein solubility and protein-protein interactions, as they can affect protein structure, function, and expression. A representative example is variants in receptor binding domain of the spike protein of SARS-CoV-2, which affect its binding to the human ACE receptor and its recognition by human antibodies. Therefore, understanding how mutations affect the changes in binding affinity is crucial for the development and optimization of a wide range of biotechnology products, including antibodies, industrial-grade enzymes, and other protein-based therapeutics and reagents. The folding and stability of proteins is critical to their function. Protein must fold into specific 3D dimensional structure to be functional. Misfolded protein can lead to loss of function. Therefore, understanding the stability of these folded structures is essential for predicting protein behavior, and binding affinity is used to measure the binding strength between PPIs. Precisely distinguish the changes in binding affinity upon mutations is an important but unsolved challenge in protein engineering. How interactions at the protein interface affect the changes in binding affinity when bound to various ligands? Is it possible to use protein fold mode to capture the backbone structure of proteins? Whether and how different types of urges enhance our predictions of mutational effects on protein-protein interactions. Here is an overview of DDAffinity. Firstly, by leveraging FoldX, we get a mutant structure from the wild-type structure of protein complex. Our model takes these structure pairs as input and derives a k-nearest neighbor graph, which is processed by future extractor and structure encoders sequentially. Secondly, by sharing the same architecture between wild type and mutant type, the structure transforms the input urge and node features to hidden embeddings, respectively. Thirdly, urge and node embeddings, as well as node skip connection, are fused by a fusion layer. Then centrality normal layer is performed residue-wise. Finally, max polling layer is applied to obtain a global complex representation. With the representations of wild type and mutant type, MSE loss is optimized to minimize the forward DDG, as well as reversal minus DDG for the sake of anti-symmetry. As shown in this figure, we designed three different neighbor residues to construct the k-nearest neighbor graph, given a central residue of mutation. The first are residues that are spatially and sequentially less than k1 in distance. The second are residues that are less than k2 in sequence distance only. The third neighbor contains only residues that are less than k3 in spatial distance. The training and validation dataset we used are derived from SCAMPy-2, which include 345 complexes and 7,805 point mutations. This dataset is generated by calculating the average mutation effect for variants reported in multiple experiments, and its 3D structures have been deposited in protein data bank. This figure illustrates the counts of mutation types in the dataset. In this paper, 5 evaluation metrics were used in data-G prediction tasks, including Pearson's correlation coefficient, Spearman's rank correlation coefficient, RMSE, MAE, and AURC, to evaluate the optimization of the human antibody task. We use hits at K, which denotes the region of top K favorable mutations in the ranked test datasets. To evaluate the performance of DDAffinity, we conducted comparison experiments using 3-fold, 5-fold, and 10-fold cross-validation with 8 baselines. DDAffinity demonstrates better performance than baselines on multiple point mutations in every metric. The prediction results are visualized below, and DDAffinity accurately forecasts the plausible variations in sidechain conformations. We also performed complex level validation on single-point mutation subsets S1131 and multiple point mutation subsets M1707 to compare the predictive performance in Table 2. Our results demonstrate that DDAffinity is able to make prediction with higher predictive performance than baselines. The performance of DDAffinity was further evaluated by performing training on the multiple point mutation dataset M3040 and non-redundant blind testing on M595 at mutation level, which were derived from SCAMP2. DDAffinity also exhibits notable improved in predictive performance compared to baselines. To evaluate the ability of our method in optimizing antibodies against the SARS-CoV-2 spike protein and in mutational scanning of the spike protein's receptor binding domain, we predicted the impact of every single point mutation on the binding affinity of the ancestral Wuhan Hu1 receptor binding domain and optimize human antibodies targeting the SARS-CoV-2 spike protein as the subject for our case study. Finally, I would like to make a conclusion of this presentation. In this paper, we proposed DDAffinity based on k-nearest neighbor residue graph, which aggregates information from spatial and sequential MPNN for accurately predicting changes in binding affinity caused by multiple point mutations. Compared with benchmark studies, DDAffinity achieves high correlation and low error performance on the data-G prediction task, which demonstrates the benefits of the proposed model in correctly capturing the local and global synergistic hypostasis property of PPIs in terms of covalent and non-covalent interactions inherent in multiple point mutations. Furthermore, DDAffinity could be well applied for the mutation efforts prediction of SARS-CoV-2 RBD variants and the optimization of human antibody against SARS-CoV-2. Well, before I finish, I want to thank my supervisor, Professor Jianxin Wang in Central South University. Thanks for your attention.
So if anyone, we can ask questions and then he will type the answer and we will read the answer. But it is what it is. So are there any questions? Go. We need the microphone.
Hi, great work. I mean, the obvious question, I don't think DDMUT PPI was in your benchmark, but it's definitely a interesting comparison after we just saw this method that also outperforms the previous state of the art. And if this is not published yet, maybe it'd be great to include it to to make the most out of it, of the benchmark.
Could you type your answer in the Q&A section?
Okay, we'll try again with the next question if there is one. And if not, I will answer for him.
So there have been a number of different methods to predict effect of mutations on protein-protein binding affinity. Now there is this new generation of methods using deep learning architectures, but it's not clear to me how actually using deep learning architectures help us in prediction, say, binding affinity, because from my knowledge, SCAMP, this leave one out cross-validation correlation coefficient 0.7, 0.8, was achieved before by previous methods. So I wonder what you think actually the advantage of using that, because also that I think for me, to me, the accuracy of prediction is not just predicting of binding affinity change, but also predicting actually of the, say, rotamer structure of the mutant. So this is basically my first question. And the second question, this reverse forward mutations, kind of people use this concept and enriching training set. So what is your prediction quality of stabilizing and destabilizing mutations?
Is the speaker able to answer? Yes. We'll include more published work in the comparison. Okay. All right. I don't know if he's typing something or if not. I apologize for the technical mistakes, but technical problems. But let's thank the speaker in case he can hear us and move on.